Hello, my name is Juliana Giles, and I am a graduate student here in the Department of Earth and Space Sciences at Columbus State University. The purpose of this presentation is to introduce my master's thesis proposal titled Understanding the Bull Creek Watershed Crayfish Community, a Distributional Survey. Although freshwater systems constitute only a really small percentage of the Earth's surface, they support approximately 10% of the entire planet's species. The expansion of impervious surface area as a direct result of urbanization has had devastating impacts on freshwater biodiversity. Streams are subject to major anthropogenic alterations like flow regulation through dams and channelization, fragmentation, habitat degradation, increased pollution, and the introduction of invasive species. All of these impacts expedite the loss of native populations and reduce the quality of many ecosystem services that these systems and their inhabitants provide us. But even though freshwater taxa are at a greater risk of extinction than many terrestrial taxa, they are more often than not underrepresented in protection and conservation efforts, as well as federal and state lists for endangered and threatened species. Many scientists have also pointed out that there's been a lack of freshwater species distributional studies, which has contributed to the current poor understanding of the factors that are driving their frightening rates of extinction. The graphics that I included on the right are from a 2015 study that was conducted by a team of researchers from the University of Wisconsin at Madison. Their study was titled Threats and Opportunities for Freshwater Conservation Under Future Land Use Change Scenarios in the United States. So the top map shows urban land cover in watersheds that have a significant freshwater biodiversity according to 2001 land cover data. And the bottom map shows projected urban land cover and biodiversity rankings in what they called their business as usual scenario. In which urban expansion continues to increase at its current rate until 2051. I'm sure that the rate of urbanization that they used has likely increased since then, since this was conducted in 2015. It's been a few years since then. But what I would like you to notice is how much dark red there is in the second map. This color indicates areas of high biodiversity that are expected to see an increase in urban land cover that's greater than 5% in just 50 years. So we know that freshwater ecosystems are characteristically diverse, but why study crayfish? Well, the southeastern United States is actually a biological hotspot for freshwater species, and it's home to nearly half of the global total of crayfish species. According to the Georgia Department of Natural Resources, and thanks to the extensive work of Dr. Horton H. Hobbs, we know that Georgia is home to roughly 70 species of crayfish, and some of those are actually endemic to the state, which means they're only found in Georgia. Crayfish play a very important role in freshwater food webs. So crayfish are shredders, which means they feed on large or coarse organic plant material. And in the process, they create small, fine organic matter that can be used by invertebrates that make a living off of collecting fine particulate organic matter. Crayfish also serve as an important protein-rich food source for larger stream-dwelling organisms like fish and other terrestrial organisms. The images that I included on the right are all from georgiabiodiversity.org, and they are just images of some common crayfish that are found in Georgia, whether they are native or invasive, like Procambarus clarki. So the goals of this project are to assess the current biodiversity of crayfish in the Columbus, Georgia area, and to gain a better understanding of how regional and local factors have influenced the distribution of the Columbus area crayfish community. The scope of this project includes the entire Bull Creek watershed because it drains almost all of Columbus. To accomplish these goals, my objectives are to conduct a comprehensive crayfish survey of the Bull Creek watershed, to collect and compare past and present land use data, and to create a spatial database of Bull Creek watershed crayfishes that can be expanded upon or used for future research. One question that I'm seeking to answer through this project is, what effect does urban land use have on the structure of the crayfish community in the Bull Creek watershed? Because urban land cover is associated with freshwater habitat disturbance and destruction, pollution of surface waters, and elevated risk for the establishment of invasives that can displace native populations, I expect that subbasins with higher percentages of impervious surface cover will have lower crayfish biodiversity than subbasins with higher percentages of more natural forested land cover. And because impervious surface cover increases longitudinally throughout the Bull Creek watershed, I expect that crayfish biodiversity is going to decrease longitudinally throughout the watershed. And just to help illustrate that, this is a map that I created in ArcGIS Pro. This is the Bull Creek watershed. So the Bull Creek headwaters are up here in this northeast corner, and Bull Creek runs all the way down through here to the Chattahoochee River. You can see, I know that there are a lot of land cover classes in the legend, but all of the red in the southwestern portion of the watershed is developed land. So as you move down through the watershed with Bull Creek, you can see that urban land cover increases immensely. And that's why I expect to see a decrease in crayfish biodiversity as I move through the watershed. Another question that I hope to answer through this study is, how has the crayfish community of the Bull Creek watershed changed over the last 40 years? Answering this question will depend on my comparison between our current crayfish survey data and crayfish survey data that was collected in the 1970s and 80s by former professor of biology, Dr. George Stanton, who spent many, many years here at CSU and is a crayfish expert. He and several of his research teams during the 70s and 80s conducted crayfish distribution surveys and studies, and many of those were conducted within the area of the Bull Creek watershed, and we have access to that data. Because a lot of Columbus has experienced extensive urbanization in the last 4 decades, I expect that the crayfish community is going to reflect these changes in impervious surface cover by being less diverse today than it was 40 years ago. Another reason that we have to believe the current crayfish community to at least be significantly different than what it was in the 70s and 80s is that we have evidence that points to the establishment of a highly invasive crayfish species, Procambarus clarki, which I pictured and mentioned in a previous slide. This is actually a video of an individual Procambarus clarki male that myself and a cohort, Dominica Sherwood, caught in Lindsay Creek, which is within the boundaries of the Bull Creek watershed. We caught this individual just this past October. And this highly invasive species has been known to displace native populations. And if it's present and successful in the Bull Creek watershed, then I expect this to have taken a toll on the biodiversity of the Bull Creek watershed crayfish community. Data collection for this project will begin in spring 2022 with a watershed scale crayfish survey. This survey is going to be time-consuming and labor-intensive. However, this is a collaborative effort between myself and Dominica Sherwood, a cohort whose master's thesis project is also constructed around the crayfish community of the Bull Creek watershed. Due to this survey's collaborative nature, Dominica and I are actively working together to locate and sample streams and impoundments for crayfish, and we are actively working out the logistics of our sampling methods. We have talked about deploying traps for crayfish. We've also talked about active sampling by using dip nets. We may opt for one or the other or a combination of the two, but that is something that we hope to have figured out very soon. My project is only going to be concerned with data collected from sites located on streams, since I'm interested in watershed scale drivers of crayfish community composition. Because I want to understand the effects of environmental factors on crayfish distribution at both the regional and local scales, I've opted to divide the Bull Creek watershed into 60 representative subbasins. This is a map of the Bull Creek watershed area that I delineated in the USGS StreamStats application along with surface water flow lines from the National Hydrography Dataset. I also included a star to represent the location of CSU's main campus, just to give you an idea of where we are at within the Bull Creek watershed. This is a working map of potential sampling sites that I have identified using ArcGIS Pro. After I created those potential sites, I used the USGS StreamStats batch delineation tool to delineate all of the individual basins for each of the sites. So all of the red dots that you see on the map represent individual sampling locations, and the area above them contained within the yellow borders are their representative subbasins. I chose to use this approach because I can use data collected from the individual sampling locations to understand local conditions within the individual basins that can influence that local area's crayfish population, and then I can synthesize all of the subbasin data to understand the regional patterns of the Bull Creek watershed crayfish community. There are many environmental characteristics that determine the suitability for crayfish presence, but because the Bull Creek watershed is encompassed within one ecoregion, there aren't really any significant changes in climate or vegetation type within my study area. Because of this, I aim to collect information about specific environmental factors that could influence whether or not a crayfish species would be present in one subbasin but not another. And urban influence is also a consideration in my choice of factors to include. Currently, I aim to collect information on water quality through a measure of sampling site dissolved oxygen, pH, conductivity, turbidity, and temperature. I also plan to collect data on the physical characteristics of the sampling locations by measuring stream width, depth, flow, and streambed composition. All of these factors are important determinants of crayfish presence or absence, but this is by no means a comprehensive list of the kinds of environmental factors that would influence whether or not a crayfish would be present in any given area. In order to compare land use changes between Dr. Stanton's past crayfish surveys and our present survey, I plan to use historical national land cover data from the 1970s and 80s, as well as the most recent data from 2019. By comparing the two data sets and coupling them with changes in the Bull Creek watershed crayfish community, I will hopefully gain a better understanding of any shifts in the community structure as a result of changes in the region's land cover. This is a map that I created in ArcGIS Pro that just shows the historic land cover data from the 1970s and 80s. And once again, this is a map that shows the present land cover data from 2019. There are a lot of steps that are going to have to happen before I can actually compare the two data sets, but this is just a visual of what I have collected so far. This project has been a long time in the making, and my hope for my remaining timeline is to hit the ground running in spring 2022. Before the end of this month, I will have assembled my final thesis committee. My proposal will be completed and defended in January of 2022. The analysis of the historical and present national land cover data will be completed by February of 2022, and the collection of crayfish survey data and environmental characteristic data will be completed throughout the months of March and April 2022. All data analysis will be completed throughout the months of April and May of next year. My thesis will be submitted during the summer semester of 2022 and will be subsequently defended during the fall semester of 2022. Finally, I will graduate in December of 2022 at the conclusion of the fall semester. And the following 3 slides are just lists of the references that I used to create this presentation. Thank you so much for your time and attention, and I look forward to your questions in class.